Hello and welcome to another episode of Momento Malum today. The common bean is an important food crop worldwide. This includes the United States, where approximately 2 million acres of dry beans were harvested in 2021. Common beans are native to the Americas, ranging from southwestern North America to the southern Andes, with the northern and southern populations forming two separate gene pools, the former being more genetically diverse than the latter. The wild form of the species was found to have unique traits not found in the domesticated populations, such as improved disease resistance. The genetic diversity within the cultivated dry bean is quite narrow, and the wild germplasm has been underutilized for increasing the genetic diversity of the cultivated beans. Several lines were bred using this process, with unique traits such as weevil resistance and root rot resistance being added into new lines. The chief diseases of common bean in the United States are Anthracnose and mosaic virus, and these can result in complete crop failure. Anthracnose resistance is conditioned by more than 20 resistance genes, some of which have multiple alleles. Of the known genes, the CO1 gene originates from the Andean gene pool and is the most studied and utilized gene. This gene also has the most variation, coming in multiple forms. These are CO11, CO12, CO13, CO14, CO15, CO1HY, CO1X, CO3, CO10, CO15, CO16, COY, COX, and CORVI. In modern times, CO12 has been overcome by a new strain of anthracnose called race 109, marking the end of the use of CO genes, as they've all been overcome by anthracnose. This has created the need for new resistance genes to bulk up the gene pool. For mosaic virus, the main method used to breed for resistance is through a single gene called an I gene, and are supplemented with other recessive genes such as BC1, BC12, BC2, BC22, BCU, BC3, and BC4. There is, however, a continuous need to further supplement the disease resistance of these populations. The result of this was a project taken on by the USDA, Midland Company, and the Michigan State University. 241 lines were collected, and these consisted of 194 wild lines and 55 landrace derivatives. These would be tested against the two most virulent anthracnose strains. These are race 2047 and race 3481. They were also tested against the most virulent strain of mosaic virus, NL3, and here are the results. But first, ad time. If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below. In total, 26 of these lines were resistant to anthracnose race 2047. 53 were resistant to race 3481, and 13 were resistant to both strains, and all of these were of pure wild stock. Overall, the land races were similar in resistance to the wild lines on average regarding race 3481, but the wild lines were on average more resistant to race 2047. Of the land races, only PI 642127, PI 63888 MXQT2, PI 638868, PI 535427, PI 317350, and PI 535435 showed consistent qualitative resistance to anthracnose. Race 3481. As for mosaic virus resistance, of the studied lines, only a few had resistance to this disease. The landrace lines 932, ARM, 174, and Giza 3 were all resistant to the mosaic virus. And the Giza 3 was also resistant to Anthracnose race 3481. The researchers conclude by mentioning that the wildlines and a portion of the land races have a great deal of resistance to Anthracnose, but very low resistance in terms of genes for mosaic virus. And all the mosaic resistance they found were in the land races, not the wild strains. And that about covers everything. Thank you for watching.